Hello and welcome back everybody to this, our last session of our Fringe Day for this year. Thank you so much. If you're joining us for the first time, welcome to the Oxford Farming Conference Fringes. And if you've been with us for a previous event, welcome back. I hope you had a really good day and learnt lots. My name is Emily Norton. I'm the chair of the Oxford Farming Conference for 2023. We're all very familiar with Zoom, but just a brief reminder, please keep your microphones on mute unless you're one of the speakers, of course. And if you have any questions, please post them in the chat. The people from UKRI who are hosting this next session will be monitoring those questions that you post in the chat, and we'll pull them forwards to post to the panel. All 7 sessions so far from today have been recorded and all will be available via the Oxford Farming Conference website after the event, and we will share those links with you tomorrow. The Fringes, of course, and the conference, which runs in Oxford, here in Oxford, from the 4th to the 6th of January, would not be possible without all of our partners and sponsors. So our thanks go to all of them, and in particular to our headline sponsor, Trinity Natural Capital Group. Thank you. I won't keep you any longer. Our next Fringe of the Day is hosted by UKRI and aligns with our 2020 theme, Farming a New Future. So it's over to you.
Thank you very much, Emily. Thank you. Welcome, everyone. Welcome to the Oxford Farming Conference Fringe Programme event, Farming a New Future, from fundamental research to real-world application. My name is Harriet Chewin, and I'm the Head of Bioscience for Sustainable Agriculture Food at UK Research and Innovation. And I work within BBSRC, which is the Biotechnology and Biological Sciences Research Council. Our role is to convene, catalyse and invest in close collaboration with others to build a thriving, inclusive research and innovation system. I'm very pleased to have been asked to chair this exciting UKRI session, examining how fundamental research can lead to practical implementation of real-world solutions that will secure our farming future. Delegates will hear how current programmes of research and innovation are seeking to provide systemic solutions to global challenges we face both today and in the future. In this session, we have 3 presentations. Each will be 10 minutes in length, followed by a question and answer session from the audience. Those questions can have, Emily's already said, those questions can be submitted electronically in the chat function, whereby my colleagues will be picking them out throughout the talks. We have a great variety of talks ahead of us. So our first presentation will be from Dr. Richard Green from Harper Adams, sharing his work on pasture-to-plate, realising the enormous potential of UK grasslands, whereby they have been looking at novel ways to maximise the food potential of UK pasture through targeted chemical processing and novel biotechnology, with the aim of making UK agriculture more resilient and sustainable. After Richard, we'll have Dr. Samantha Cook from Rothamsted Research, sharing her work on integrated pest management for oilseed rape. She is working towards the development of ecologically based tactics for integrated pest management that will help our farmers to sustainably intensify production while minimising negative environmental impacts. The final presentation will be from Professor Rod Wilson from Exeter University, sharing his work on transformational blueprint for a blue economy on UK terrestrial farms, integrating sustainable shrimp production in a changing agricultural landscape. This project aims to demonstrate the social economic benefits of a world-leading terrestrial blue economy, contributing multiple public goods to reform UK agriculture. So without further ado, I'm going to pass over to Richard. Richard, you have 10 minutes and I'll let you know when you have 2 minutes left for your presentation. Thank you, and over to you, Richard.
Thank you, Harriet. My name is Dr. Richard Green. I'm from Harper Adams University. I'm here to tell you about our P2P Pasture-to-plate project that we're running in conjunction with the University of Bath. So what was the background to the project? The background is really, we wanted to look at how we could improve UK food production At the moment, we've got a current system which we don't believe is long-term sustainable. So it's intensive, it's got a lot of inputs, it's high maintenance, and but it does have the great advantage of actually producing a lot of food. There are a lot of ideas floating around at the moment about how you can change the farming systems to make it more environmentally sustainable. Most of these are sort of green-based ideas, and they're very eco-friendly. So if you look over on the left-hand side of this slide, you can see some of those ideas. I mean, most of these systems end up being low intensity, low input, low maintenance. They tend to be very environmentally friendly and sustainable. They also tend to be very resilient. The problem with them is that the elephant in the living room is that they actually are very much less productive. They produce less food. There's much less farm income, and there are fewer farming jobs. What the government really wants us to try and do is to do all the things on the right-hand side of this slide. The key one being to increase food production, not reduce food production. So what we did was we went back and we thought fundamentally about the way food is produced, and we looked for a resource in the UK that is underutilised. The resource that we decided to look at was grass. It makes up 70% of UK farmland. At the moment, the only way we can use it is by feeding it to animals, and then those animals turn it into milk or into meat, which we can consume. And they also have a byproduct of fertilizer that we can put back onto the grassland. The problem is this is a very inefficient way of actually producing food from grass. It takes 15 to 25 kilograms of grass food, the potential food that's in grass, to produce 1 kilogram of meat. This isn't really a good return on the use of grass. So we asked ourselves, could we do it in a different way and increase the output from the grass? So what we came up with the pasture-to-plate concept. The pasture-to-plate concept takes grass, and then we process it in a processing plant. And I'll go through the process in a minute, but we basically fractionate the grass, break it down using novel yeast biotechnology. And then with those food fractions, we can then incorporate those into food. So we produce a whole range of different food fractions. These include omega-3 lipids, proteins, oils, fats, carbohydrates, fiber, and vitamins and minerals. The great thing about this process is that it only takes 1.5 to 2 kilograms of grass food fractions to produce 1 kilogram of edible food fractions. So how does the process work? Well, we take the grass and then we feed it into a mechanochemical conversion process, and then we can break it. So we break the grass down, we remove the lignin from the grass, and then we can remove the soluble edibles, the vitamins and carbs, and the protein fraction. We can then go through a second process and remove the lipids. These are basically oils that we can use for cooking. We then feed yeast on the remaining material. This yeast produces carbon dioxide. The leftover material goes off to be fertilized, but it produces a load of yeast biomass, which is made up of lipids, proteins, vitamins, and carbohydrates, which we can then feed back into the start of the process so we can extract those. The waste heat and the waste CO2 from the process we intend to use to put into greenhouses to help increase the amount of greenhouse production. So what are the benefits of doing this? Well, it is a significant, potentially, it could be a significant increase in UK food production. And we're looking at 25 to 50% increase, with us potentially making the UK self-sustaining in food. It will increase UK resilience to climate change because grass is a much more resilient crop than any of our other crops. It's more resilient to flooding and extreme weather conditions and recovers more quickly. Therefore, it makes our farming more resilient. One of the big outputs from this is that we potentially could produce substitutes for things like palm oil and soya protein. We believe we could produce enough oil very easily, not only to have enough to serve all of our own internal UK needs, but also to export the oil as well. That's one of the great benefits of this system. It also has, because we're replacing palm oil and we're replacing a lot of foods, a lot of ingredients are imported into the UK, it has environmental benefits. It reduces food miles. It also has the potential to allow us not to destroy the rainforests to produce food, so we can actually produce foods here and in Northern Europe. It will provide a fantastic new opportunity for grassland farmers. We aren't going to steal your grass. This isn't a matter of, oh, I keep cows on my grass. We've worked out there's enough unutilized grass in the UK for this whole process to work. In addition, for farmers, we will be paying a good price for that grass. So it's a lucrative new market that you can get into. And as the produce is vegan friendly, it allows farmers who've been closed off to that vegan market to actually produce produce that they can sell to vegans. It will provide a commercially viable non-livestock based market for forage production. What this means is that essentially we can now put grass economically into the arable rotation. This has some huge advantages. One of the best ways of tackling black grass that is resistant to chemicals is actually to put a crop of grass in and then harvest the grass, and you can take out a lot of the black grass before it actually gets to the point of seeding. This helps you reduce your black grass population. So it's a way of including economically grass in an arable rotation. It also, there's a lot of land has been put over to arable production, which should never really have been put into arable production because there isn't anything else for them to do with it. The challenge that you've got there is that quite a lot of this land is low-lying, it floods, and we've got fields around here which flood 2 or 3 times a year, and the farmers replanting that seed, reseeding those areas 2 or 3 times in a year, which just simply cannot be economically viable. The great thing about this whole process is it doesn't require any new farming machinery. All the farming machinery we need to do this is there. It's already being used to produce forage. It's already being used to produce silage, and those are the ingredients we'll be putting into this system. In addition, the bioprocessing technology is mature technology, so it's not going to take years and years for this to come through to the market. We're looking realistically at having grass-based food products in the market within 3 years. So before we finish this project, we're hoping to have grass-based food products ready and having to start to be building that new market for farmers who want to sell grass to us. It's a massive opportunity for investment in new UK food industries, and it's a massive opportunity for companies to develop new markets. What I want to make is really clear is that we're not talking about producing some sort of green gunk. What we're talking about is the things that we're going to be producing are substitutes for things that are already being imported. So they're base ingredients in a lot of the foods that you already eat. We're not changing what you eat. What you eat will be pretty much exactly the same as it was before. It's just that the ingredients that will be going into will be produced by UK farmers rather than being imported from abroad. Okay. I'd just like to wrap up there. These are some of the partners that we have in the programme. A mixture, it's a very broad, eclectic mix of different people, all working together to try and make this a reality. So thank you, Harriet, and I'll pass back to you.
Thank you very much, Richard, and thank you for your talk. As I said at the beginning, we're going to take questions at the end, so please post them in the chat if you have questions specific to Richard's talk, but we will take them at the end. So without further ado, I'll pass it over to Sam. I'll say again, because I just 10 minutes, please, and I'll let you know when you have a 2-minute warning. Thank you, Sam.
Okay, thanks very much. And can you see my presentation first of all? Okay.
Yeah.
Oh, I'm on the wrong side. Hang on. Swap. Sorry, this is right. Okay.
There you go.
I'm Sam Cook from Muffin Research, and I'm going to be talking about integrated pest management strategies for cabbage stem flea beetle and oilseed rape. So first of all, what is integrated pest management? There are a lot of different explanations about that out there, but I like the one from the US Environmental Protection Agency, which describes it as an environmentally sensitive approach to pest management that relies on a combination of practices, which includes the judicious use of pesticides. And it uses lots of information on the life cycles of pests and their interactions with the environment. And the EU produced the Sustainable Use of Directives back in 2009, which aimed to achieve sustainable use of pesticides in the EU. So every EU country, including the UK as well, has a responsibility to set rules for farmers to follow to ensure that pesticides are used judiciously. But they also included in this directive promotion of the use of integrated pest management and alternatives to pesticides, which can be thought about in terms of the pesticide triangle, where you've got cultural control methods at the bottom, including resistant or tolerant cultivars, or use of enhancement of natural enemies and antagonists to control pests naturally. Then you think about diagnostics. Have you got the pest, decision support, and use of thresholds in terms of risk assessment? Then if you realise you've got a problem, using biological controls first, if they're available, and using chemical or synthetic chemicals as a last resort. IPM's really moving up the agenda now, and a lot of farmers are now encouraged to fill in an IPM plan to show that they're doing it. I've been working on developing integrated pest management strategies to roll out to farmers for many years. I work on oilseed rape because it's got a lot of pests to worry about, but most of our work has focused on cabbage stem flea beetle and the pollen beetle, and I'm going to talk about flea beetle in this session today. I like to say you get two pests for the price of one, because both the adult and the larvae cause damage to the crop. The adult feeding chews holes in the leaves, and this can threaten establishment. The larval tunnel the plant, eating it from the inside out, and they weaken it and causes susceptibility to disease. The adults were previously controlled by nicotinoid insecticides, and the larvae were previously controlled by spraying pyrethroid insecticides. But as you know, the UK and EU banned the use of neonicotinoids back in 2013. And we saw lots and lots of bare ground fields that should have had oilseed rape in them. This wasn't just due to the banning of neonicotinoids. Unfortunately, farmers faced a double whammy, as that year really became obvious that the cabbage stem flea beetle had become resistant to pyrethroid insecticides, which were the only other alternative control available to them. And this was quite prevalent across the UK. So farmers are in a position where really they've got nothing left to control this pest. And this has led to a really massive drop in decline of the oilseed rape cropping in the UK, over a 46% decline since its peak in 2012. And this has led to the need for us to import far less sustainably produced alternatives. So we really need to get oilseed rape back in the rotation. So let's start at the bottom. I'm going to talk mainly about the development of resistant or tolerant cultivars, which is a keystone corner to IPM strategies. But unfortunately, resistant cultivars aren't available for any insect pest of oilseed rape. So a few years ago, I ran a study with my colleague at Rothamsted, and we screened a diversity set from Origin, and we looked at the assessment of flea beetle damage in the field, and then we tested those same plants in the lab to compare cabbage stem flea beetle feeding. And you see from this little graph at the bottom there, we ran 5 replicates, and we saw a variation in the amount of feeding damage between those different varieties, with some of them having absolutely no feeding at all, and others receiving quite a lot of feeding. So there is genetic variation out in the Brassicaceae. Rachel Wells at John Innes also ran some similar studies. And so with UKRI funding, we were able to combine forces and begin a new project called Breeding for Resistance to Cabbage Stem Flea Beetle, which started last year. So this is this project, a toolkit for breeding resistance to adult and larval stages of flea beetle. It's got three main objectives. The first is to explore the genetics of this plant resistance that was seen in the lab. And we're going to be testing other members of the Brassicaceae, Arabidopsis, turnip grapes, and oilseed rape mutants to try to confirm these candidate genes and the way they function in providing the resistance that we've seen in our lab studies. Then we would like to explain what the differences are in adult palatability and how that's produced. So we're going to benchmark commercial varieties of oilseed rape by comparing their performance in adult feeding trials against those that we found in our lab studies. So maybe we're going to determine if the defense mechanisms are due to the hairs that are present on the stems, for example, or is it the glucosinolates, or is it the cuticular waxes? What's driving these differences that we're seeing in the field? Also, in objective 3, we want to understand the tolerance of some varieties that we've seen to cabbage stem flea beetle larval attack. So we're going to be, again, performing field trials with a wide range of commercial materials from our collaborators and try to identify the genetic basis and the mechanisms between these differences. We're really lucky that we have got a very wide range of breeding partners. So this project will really be able to pass the information out to breeders as soon as possible and get those genes into commercial varieties for growers to use as soon as possible. So field trials, we're now in our second year of field trials. We have tested so far 20 lines and we've replicated those each 5 times in the field at Rothamsted and John Innes. And here's a picture of the difference that we can see very easily simply in flowering phenology. So we're seeing lots of differences in these field trials. We are looking at the adult feeding damage, so taking the cotyledons from the plants, scanning them and using image analysis to identify the amount of leaf area eaten by the flea beetles. And here you can see some preliminary data, caveat preliminary data. You can see at the bottom there that some 2 or 3 lines have a much lower mean area of eaten compared to other lines. So we're quite happy that we're seeing differences here. In terms of larval antibiosis and the ability of the plant to either kill the larvae that are infesting it or reduce the numbers that are developing, we're having to screen lots and lots of varieties, and we've developed a neat way of doing this. So we put the plants on chicken wire and let the larvae drop down, and we count the number of larvae that end up in the pots at the end after about a week. We're really quite happy that we've developed a really novel way of doing this, and we're very confident that the number of larvae that we find in the pots at the end of the experiment is related very closely to the number that we would have found if we'd dissected it by conventional methods. So it's much easier and just as accurate to do it this way. And again, we have found some nice differences in the materials that we've tested. So you can see there at the right-hand side, these 2 cultivars had much lower larval infestation per gram of the material that we tested. Lastly, I'd like to move on to the other corner of cultural biocontrols, and that's the enhancement of natural enemies and antagonistics. So how can farmers farm and use their management practices to increase the amount of natural enemies that are going to provide pest control for free? My PhD student, who's now postdoc at Rothamsted, has spent some time working on Microctinus brassicae. It is the adult parasitoid of the adult stage, so we're very excited about this, but very little is known about it. So Patri wanted to know how prevalent it is across the UK and what the parasitism rates are like. So she asked farmers for lots of samples. We're really, really happy that we received over 200 samples from across the UK. She was able to demonstrate that the parasitoid is found in over 70% of the fields that she studied, and the parasitism rates were ranged between 1 and 30%. So 30% is a really, really good biocontrol number. So we could actually start to rely on biocontrol from this parasitic wasp. But in our future studies that will be funded by UKRI in our Growing Health Institute strategic programme, we're hoping to use and develop molecular diagnostic tools to quantify parasitism and then use that to understand the influences of management. Some farmers are undersowing, for example, or hedgerows, and the role of landscape in changing the parasitism picture across the UK. So I think these two projects really show that together with UKRI funding and research institutes, farmer involvement and industry stakeholders, together we can fight the flea beetle and deliver practical tools for next generation integrated pest management so that oilseed rape can be a productive and sustainable part of the arable rotation in the future. Thank you very much.
Thank you very much, Sam, and thank you very much to both the speakers for keeping on time. It's really helpful. Thank you. And I believe our next speaker is here. I was slightly panicked, Rod, but I believe you are here now. So, Rod, I will pass over to you. Welcome.
Thank you very much. I need to share my screen. So can you confirm that you can see my slides?
Yeah, we can see them. Thank you.
Excellent. Thank you very much for inviting me to present here. So my name is Rod Wilson. I'm an animal biologist at Exeter University. And I'm here to talk about this new project that started just in the last few months with this rather lengthy title, which is really all about integrating aquaculture, which is growing seafood essentially in indoor systems, but in this case, integrating it alongside terrestrial farming practices in the UK. And specifically, this project is focusing on a species known as king prawns in the UK. And because they're tropical, we need a source of heat if we're going to grow them in the UK. And it's all about how we can do that sustainably here, better than where it's done overseas. So the species in question is the highly desired king prawns, which the British public already likes a lot, and it's a very healthy seafood. And we currently import something along the lines of 319 million pounds worth from places like Southeast Asia and Central America. There is an issue with the way it's farmed in some of those countries, and for some reason, there you go. By the way, we've shortened the title now to a much easier to remember UK Sustainable King Prawn Project. The way it's often farmed in those countries overseas that have the right temperatures ambiently is along coasts where there would normally be mangrove forests, which are fantastic for removing CO2 and for protecting the coastline from high water levels. So when you cut those down and instead build these outdoor ponds for growing your prawns in, it's actually environmentally having quite a negative effect. The other thing that's often negative about it is the use of chemicals like pesticides and antibiotics, which is something we would definitely want to avoid. But there is another way of doing it, and we can do this within the UK or any colder country, as long as you can provide a reasonably cheap and sustainable source of heat. So it turns out, as many of you know, that many farms in the UK utilize anaerobic digesters to take farm wastes and produce biogas that can then be transported at long distances and sold, or burned on site to create renewable electricity. And again, that can be transported over long distances. However, there's also an awful lot of heat created by these anaerobic digesters, both within the burners to produce electricity and from the anaerobic digestion process itself. And that waste heat is currently literally wasted, goes up into the atmosphere and is gone, which is not ideal. But the problem is you can't move heat over large distances. However, if you can put something that needs that heat next door, then you're onto a winner. Now, with tropical species of king prawn, for example, we have a footprint of about 10 by 30 meters for a single unit of prawn production, which can very easily be housed next door to anaerobic digesters and utilize this waste heat to do pretty much all the heating you need for that tropical species to live and grow in. And being quite small, you can scale up as much as the size of your anaerobic digester will allow. So with this in place, you end up with about every 3 months, you can harvest and produce the high value, healthy seafood that king prawns represent. grown locally, and therefore you'll get the freshest supply to the UK consumers as well. Interestingly, most people don't realize that king prawns, because they molt to grow like other crustaceans, crabs and lobsters, they shed their exoskeleton, which contains a lot of things that we normally think of as waste. But in fact, the chitin in their exoskeleton has a value because you can convert it to something called chitosan, which the pharmaceutical industry really wants to get hold of and will pay very good money for. So there's actually two high value products that come out of prawn farming. One is the prawns themselves in our supermarkets. The other one is the chitin, which forms chitosan that's used in the pharma industry. Now, if we're going to grow prawns sustainably, we've got to make sure that all the wastes have a proper place to and usefulness. So there are both water waste and solid wastes that come out of prawn farming that we're going to be testing as part of this work with colleagues at Rothamsted University to see whether they can be used as a soil enhancer to help crop production. And if that's not the case for whatever reason, then there's always the option of putting the shrimp waste or the prawn waste back into the anaerobic digester to generate more gas and more electricity. So that's another area of science that we're going to be doing as part of this project. So we have 11 academics involved across Exeter University, Reading University and Rothamsted, and we have 10 project partners, including seafood retailers, aquaculture, nutrition companies, renewable energy companies, etc. And we've broken down the project into 4 work packages, which I'll talk a bit more detail about in the next few slides. The first one involves social science colleagues at Exeter and Reading. They're essentially going to do the modelling for understanding the true economic viability of doing this in the UK at a commercial scale, but not just the economics of it, the environmental benefits that would prevail as well, which would include things that would benefit from the new Agricultural Act, whereby incentivizing farmers to include prawn farming on their land would mean they can do other things with their money that would provide, other things with their land that would provide public good for public money. And that would include using other land for reforestation and removing more CO2, improving biodiversity, opening up more land for recreation as well. And don't forget that by doing this, you're enhancing or boosting the farm-based renewable energy sector as well. So the two have an interplay there. So that's work package 1. Work package 2 is much more about making sure that the conditions we grow prawns in these indoor systems in the UK are optimal for the health of the prawns and from their growth rates, so that the yield you get is maximal. And that's yet to be optimized for prawns, but that's something we're working hard on as biologists. The third work package is simply a proof of concept, whereby we've got an anaerobic digester site near where we work here at Exeter University, a few miles away, where we're going to build a small-scale prawn farm, and in particular use that to generate the kind of waste that we can test the full circularity of those wastes, as I described previously. And then the final work package is all about, if we really do scale up and start to produce a significant proportion of the king prawn that we eat in the UK instead of importing it, and we reckon conservatively that this could represent at least a quarter of that £320 million worth that we currently import. If we're really going to do that properly, we need to have our own.
Because I'm writing to chat to him repeatedly about, interpreting function, everything, and I'm speaking in all channels, but he just doesn't react. Sorry.
Rod, I don't think that's aimed at you. I think some of them maybe have taken their microphone off mute. So please carry on. Thank you.
Okay. So if we're going to do this sustainably, currently the only prawns we can bring in to start a production cycle are the larvae that would come from places like Texas. Now, the UK really needs to become independent of those overseas suppliers because of reasons of disasters that have knocked out those hatcheries in the past. So we can't afford to do that within this project, but we do want to create a scoping team to look at a business case, or create a business case to understand what would a UK hatchery type system require to be built around it in terms of infrastructure and the right people involved. So in a nutshell, that's our project. We have an advisory board we put together who we hope will keep us on the right track of thinking always about what's the most relevant for farming systems in the UK and why things would and won't work. So this is the list of people we've got representing DEFRA, CFAS, carbon monitoring, leaf, for example, and farming practice as well. And I think that's me done. Thank you very much.
Thank you very much, Rod. I think we go to, thank you, not sharing your screen. Thank you very much to both to Richard, Samantha and Rod for your presentations. We've had a few questions. I've had a few questions directly in the chat, so we'll start that off. But please, if there are any more questions, please pop them in the chat and we'll pick them up as we go through. So we have 10 minutes. The first one I had was quite an open question, which is really around how do you see research and innovation shaping the future of farming? Perhaps thinking outside of the areas that you've talked about today. I don't know if anyone wants to pick that up first, or I'll point to you. I think it's one for everyone. I'm going to pick on you because I can't see a volunteer. So I'm going to go with Richard first, please.
I think the way we're looking at it is, if we're going to produce enough food, we're going to have to think outside the box. We can't carry on doing exactly what we're doing now. We've got to find new ways of doing things, and we've got to rethink the whole process. So I think farming is going to change. It's going to see a lot of change in the next few decades. It's going to have to if we're going to feed people. And the challenges are going to be not just from the UK, but globally, as it becomes more and more difficult to grow food on other pieces of the world, other pieces of land around the world.
Thank you, Richard. I don't know, with Sam, would you like to come in on that question as well?
Well, just to echo that, really, obviously, yeah, research and innovation is going to be key to move forward. We're now really moving into an era where we probably won't have pesticides in the way that we know them now. We're moving, the EU are banning them. We're finally, as a society, recognizing that that's not the way that, we don't want them in our food anymore. So how can farmers farm for food without using pesticides, but without reducing their output quite as much as maybe organic farming does? I think we're going to see much more robotics, and the use of sensors to optimize these systems is going to be incredible in the future. So there's a lot of work to be done to enable production to feed everybody, but to maintain a healthy ecosystem as well.
Thank you, Sam. Rod?
Yeah, I was going to say two things. So in terms of aquaculture, which I know most about, the United Nations tell us that we need to double aquaculture production between now and 2050 to meet the projected demand. We're only going to do that by doing things differently to how we do it today, because there isn't the space to do it along the coastlines all the time. So we've got to think differently. And in terms of how research influences that, I've got personal examples over many years of it working in two directions. We get fish farmers, for example, coming to us and say, we've got this problem. This crops up time and time again. Is this something you can help us with? So we get the farmers themselves asking us to, as scientists, to try and solve problems they know already exist. But the other direction is we go and visit farms and we go, oh, I didn't know you did it that way. Did you know about this? The thing that we know as kind of scientists who do things just out of interest. And they go, no idea, hadn't heard about that. How will that influence us? And we'll say, well, it could easily do this, this, and this in terms of improving growth rate or health of your animals and resistance to disease. So I've got plenty of examples of a very two-way relationship in research being directed in both ways to help with food production.
Thank you. Thank you very much. Thanks to all the members of the panel. I've got one here, which is, I think, most directed at you, Richard, which is the expectation that the grass harvesting and processing system will be principally on lowland arable farms, or are we thinking if the farm currently harvests grass on upland farms, this could be a possibility? And are there any environmental issues to consider, e.g. higher nitrogen applications?
The answer is yes, we are looking at all farms. Anywhere where we can harvest grass is possible to be included in this system. There are a lot of logistics challenges. It's about where we start. A lot of the project I didn't have a chance to talk about is We're looking at how we're going to site these plants, how big these plants have to be, and then it's how we get the grass to them as easily as possible. The second part of the question was on...
Are there any environmental issues to consider?
We're trying to use, we're hopeful, and we probably will have to use some fertilizers, but we're hopeful that the waste product from this process will provide the fertilizer. Now that's a bit of wishful thinking there, but environmental challenges, in actual fact, we think we've probably got environmental benefits from this process, because we're looking at, as part of the project, we're looking at increasing the biodiversity of grasslands as well. So we're looking to put a lot more flora back into the grasslands, and to see if that's going to have an impact on the process. So we're looking at both. We're looking at arable and upland farming areas, anywhere we can harvest grass. We're looking at including grass in arable rotations. So we think there's just massive potential here.
Thank you very much, Richard. The next question I've had from the delegates is around how do you obtain and develop your research partnerships with your relevant partners and organisations? So really, from your experience, how did these come to life and how did they come to fruition, I suppose, with the project partners within the programmes you've talked about today, or how you do it with other programmes as well? I'm going to point to Sam first, if that's okay.
Yeah, well, in my particular case, yeah, I've had a long history of working with industrial collaborators, and at first it was the project that drove that. So I had an idea that I could see that there was an industry need for a more sustainable and a better monitoring trap. So that was my idea, and I brought together some industry partners that I felt could help with that. But more recently, the project that I talked about to develop the flea beetle resistant plant cultivars, that mainly came from a relationship that I already kind of developed over time with Rachel Wells at John Innes, who's leading that project. And because it's so focused on Aussie grape, the breeders, the UK breeders of Aussie grape were natural collaborators to have in that project. So I think that it was kind of obvious, but the whole collaborator researcher. So so we've got, to answer your question, yes, we've got UKRI researchers involved and academics. So we've got a three-way partnership there, which has been developed over time.
Thank you very much, Sam. Rod, would you like to come in on that question as well?
Yeah, there's a number of different ways that those interactions with industry happen. I think the first time I was involved in any aquaculture, fish farming related research, I basically had an idea to do with the fundamental biology of how fish digest their meal. And I thought, I think this is a good idea where we can improve the efficiency of their digestion and therefore make them grow faster. Surely an aquaculture nutrition company should be interested in this. So I sought out companies to figure out whether they were interested. They were. We got funding, we did some research. Since then, that happens as well, but often I get the farming community coming to me and saying, we've heard you're a fish biologist that knows this, that, and that, and the other. Can you help us with this problem? And then occasionally, like the project I just described to you, I already had the connections with the aquaculture companies and the nutrition companies, but because this project involves renewable energy, and the seafood that's really popular in our supermarkets, we thought we kind of need retailers, and that's how we got Sainsbury's and Line Seafood involved. But I had to seek them out and say, who's a good person in those companies I should contact? Got in touch, and they immediately said, we're interested. And the renewable energy company said, we're interested, because they've been wanting to find a use for their waste heat for a long, long time. So they're very pleased to be involved. So it works in many, many ways, I guess, is the answer for me.
Thank you very much, Rod. And I guess I'll turn to you, Richard, in case you want to answer that as well, is kind of thinking about the audience online. If they wanted to get involved in a programme, how best do they link with academics like yourself?
Very similar way to what Wilson and what Rod and Sam have said. With this particular project, we started with our network of partners that we'd already, and we get, like Rob, we get approached by a lot of different people. But we started with our network of partners and they put us in touch with other people. But I spent an entire summer, I identified companies that we wanted to work with. And you don't start at the bottom, start at the top, go for the managing director, go for the highest person in the company, get in touch with them. You'll be surprised, they will reply. If you pitch a good idea, I put together a really nice presentation, explained to them what we were trying to do. Then I pitched it straight to the person at the top. Go to the top, ask them, they'll put you down, they'll push it down, but they'll make sure someone's listening to what you're saying. And that's how we got all of those partners. And we've got most of the major oil production companies in the world, food oil production companies who are partners in this project. So yeah, don't be frightened to ask people to be partners in your project, and don't be, if you're going to ask people, ask the person at the top. Don't start with the guy at the bottom and try and work your way up.
Thank you very much, Richard. I think the next question is actually aimed at me, and I was just hoping I was chairing the session, which was aimed as, does UKRI support soil health projects? So we've had a number of initiatives in this area, in the area of soil health over recent years. Some of them have been looking domestically, and some have been internationally. The last one was EJP Soils, which was a European programme. There will be others hopefully coming down the pipeline, and also soil health is very much within BBSRC's remit. It's also within NERC's remit, and we have open calls always available to apply to as well. So please look out for those. Please look out for on the website, particularly in the UKRI funding finder, is where you'll find those initiatives. I'm just checking if there's any more questions. I've got one here, which is, with the growth in vegan and vegetarian diets, is the waste heat from the anaerobic digesters being used to grow fruit and vegetables all year round to reduce imports? I think that's probably aimed at you, Richard.
There is a challenge with anaerobic digesters, that the amount of waste heat depends on the type of what you're actually putting into the anaerobic digester. Some anaerobic digesters you need to put heat in, not just get heat out. So you have to be a little bit careful about saying that all anaerobic digesters create lots and lots of heat. It's not necessarily correct. We know that yes, I mean, British Sugar are a classic case of that their sugar processing factories produce a lot of heat, and they put a lot of glass houses in. I think in terms of what we're proposing in terms of growing the yeast on it, that will produce heat, and we're looking to use that heat to go into glass houses along with the carbon dioxide, which is an added benefit. So yeah, I hope that answers your question.
Well, thank you very much, Richard, and thank you again to all speakers, to Rod, Sam and Richard for great presentations and for answering all the questions from the panel, from the delegates today. So thank you, and thank you to those coming online as well for those questions and taking part in today's session. I'll pass back over to Emily. Thank you.
Harriet, thank you so much. That brilliant set of presentations. We have had the most fantastic day. I'm not going to take your time now for any length at all, other than to say thank you and to repeat again those messages around the videos have all been taken today. They will all be shared with you tomorrow. Huge thanks go to our headline sponsor, Trinity Natural Capital Group, for making this day free and available globally to our audience of viewers today. Thank you so much for taking part, for staying with us and for answering and posing some great questions to our Fringe hosts today. Particular thanks to our Fringe hosts for taking the time to set up and arrange these calls for everybody. And my sincere thanks goes to the inimitable Marcus, who sat here in the background dealing with all of the tech challenges and keeping the whole show on the road. So thank you to him and his team at Hawk for putting it all together. And thanks also to the service providers who've really pulled together to make sure everybody's represented today. Thank you. We can't wait to see you in Oxford in just 4 weeks' time. It's going to be a brilliant event. I just cannot wait just to be here with everybody else and enjoy all of the atmosphere that the Oxford Farming Conference has to offer. I'll leave it with you. Thank you and good night.